Hello, my name is Katie Bottenhorn, and today I am here to talk to you about conventional and automated neuroimaging meta-analyses. So a little overview of what I'm going to talk about today. First, we'll start with the broad strokes of what the difference between conventional and automated meta-analyses are. And then we will go through each manual and automated meta-analyses separately, including the process, some examples, and pros and cons of each approach. And then we'll talk about semi-automated meta-analyses, which really is the best of both worlds. And finally, we'll recap with a little bit more information on how to take which approach. So what I'm hoping that you will gain from this talk is first an understanding of the differences between conventional and automated meta-analyses, along with some strengths and weaknesses of each approach and when you might use one over the other. And finally, the beauty of semi-automated methods. We're going to use the example of NeuroSynth Compose, which is also going to be a little teaser for the tutorial section of the course, as that is the platform we are going to be using. So in the broad scope of meta-analysis, today we're going to talk about the literature search process, data curation, and data extraction. And if you're interested in a little bit more information about the best practices for neuroimaging meta-analysis, I definitely recommend this 10 Simple Rules paper. And because our process of searching, curating, and extracting does depend somewhat on our research question, I'll address a little bit of point one also. And then in the next talk, you'll hear about Kendra, and she'll talk a little bit more about the further steps in the neuroimaging meta-analysis. So the difference between manual and automated methods is really in the process, of course. So for a manual meta-analysis, you're going to go ahead and search through a publication database like PubMed to find papers from meta-analyzing. And then you're going to look through each of those papers. And in the case of a coordinate-based meta-analysis, you'll extract the brain activation coordinates. In the case of an image-based meta-analysis, you might email the authors to request those images if they haven't already been shared. Spoiler alert, share your maps. And then you might annotate each paper. On the other hand, in an automated meta-analysis, you're going to be searching a neuroimaging meta-analytic database to find already extracted results, and then you'll just meta-analyze them. So you get to skip a lot of steps. A little bit more information on what those steps are. In a manual or conventional meta-analysis, first you are, of course, going to have to find the papers worth meta-analyzing. So you're going to search a publication database, like I mentioned earlier, PubMed, Web of Science, PsycInfo, etc. And then from those search results, you're going to identify the candidate studies for your meta-analysis, because not all of your search results will be worth including. And then from those candidate studies, you will collect metadata and activation coordinates from the paper results. And you might annotate by adding a couple or a little bit of additional information about each of those candidate studies. Some examples of previously done manual meta-analyses include a meta-analysis of executive function in schizophrenia, a theory-driven meta-analysis looking at a hierarchical model of social cognition, a meta-analysis of working memory that identified both a core network and component networks that contribute to different aspects of working memory, and a follow-up to that meta-analysis a few years later that focused specifically on verbal working memory and also included papers that had been published in the meantime. So those last two are an example of something that Alejandro mentioned in his talk, which is that it is worth repeating a meta-analysis for lots of reasons. Here we have two displayed, one of which is that there were definitely working memory papers published between 2012 and 2019. And so in updating a meta-analysis, they can include more recent results while also or focusing on a specific aspect of a meta-analysis. So let's go through some of the limitations of the manual meta-analytic process. And to do this, we're going to be using the example of a meta-analysis that I published a few years ago that looked specifically at the brain networks engaged during naturalistic fMRI paradigms. So this is things like watching movies, playing video games, and navigating virtual reality environments in the scanner. And first, we're going to talk about the search process. So when you are searching a large publication database, you might get a whole bunch of non-neuroimaging papers in your results. For example, in doing this naturalistic meta-analysis, my initial searches returned 754 papers. And of those 754, I included about 110 in the actual meta-analysis. So that brings us to the next step, which is curation. And curation takes time. So narrowing down those 754 experiments to 110 worth meta-analyzing included several steps of screening these papers and determining their eligibility. And it's very time consuming. And then on top of that, the process of extracting coordinates for a coordinate-based meta-analysis is also pretty tedious and time consuming. So imagine from these 110 papers, I had about 380 individual experimental contrasts. And if, for example, each of those had about 10 activation coordinates, that means I'm typing in 3,000 XYZ sets of brain activation coordinates into a spreadsheet. It is tedious and time consuming. And finally, if you are adding additional annotations to each of those studies, that takes time too. So in this case, I was annotating each study for the type of paradigm it used, film, virtual reality, video games, etc., because I wanted to see how each of those stimulus types was represented across the networks that we identified. So the overview, the big picture limitations is that it just takes time and energy. If you had unlimited time and unlimited energy, anything is possible with manual meta-analysis. But if you have unlimited time and unlimited energy, you should really let me know. So why would we bother using painstaking, time-consuming meta-analytic methods the manual way when we have automated options? And that is largely because the data available in large databases are often insufficient for our research questions. So large-scale databases are often populated with an automatic algorithm, and automatically extracted data can be pretty noisy. The keywords included in meta-analytic databases might not cover the concepts that you're interested in meta-analyzing. For example, in the case of the naturalistic meta-analysis, I think I did try searching neurosynth, and I think I ended up with 20 papers, so didn't quite cut it for a meta-analysis. And finally, several of the large databases available right now do not distinguish between brain activations and deactivations, which can further add noise to your meta-analysis. So here we'll go with a tale of two meta-analyses, both looking at social cognition, but in slightly different ways. The first is a manual meta-analysis of the research domain criteria social constructs from the NIH. And the second is a broad social meta-analysis. So in the case of the manual meta-analysis, we had about 239 papers, whereas in the automated meta-analysis, they had about 1,000. And this is largely because the research domain criteria meta-analysis was really interested in looking at papers that used experiments analyzing affiliation and attachment, social cognition or social communication, perception and understanding of self and others, which are the RDoC constructs. And we wanted to know specifically about the individual brain activations underlying each of these aspects of social cognition, in addition to overall social cognition. On the other hand, this automated meta-analysis looked broadly at social cognition. You can see those meta-analytic results in yellow, and they combined that with data from a functional MRI task that was looking at social cognition, shown here in blue, and you can see the overlap in green. So same construct, different questions, different methods. And now let's talk about automated meta-analyses. So here, the main difference is that you get to skip a lot of steps. So when you're doing an automated or large-scale meta-analysis, you're going to be identifying studies from a meta-analytic database instead of a large publication database. And these neuroimaging-specific meta-analytic databases only include neuroimaging papers. So that cuts down a lot of time for the curation process and the extraction process. And in the case of an automated meta-analysis, you're probably not going to be doing a whole lot of annotation. So that's a lot of time saved. There are several platforms available for doing automated meta-analyses. The first of which, released in the early 2000s, is BrainMap. BrainMap is a manually populated database of brain activation and voxel-based morphometry coordinates from published papers. It currently includes over 4,000 papers, or over 4,000 functional papers, along with some very rich metadata describing the experimental context, the participants in the study, paradigms, etc. And this is possible because by manually populated, what we mean is that an actual human being read each of those 4,000 papers and used their human brains to extract the relevant information regarding the study results and what the study was about. The next platform on the scene, about 10 years later, was Neurosynth. Neurosynth, on the other hand, is an automatically populated database of brain activation coordinates from published papers. It currently has over 14,000 papers representing functional imaging studies. And it includes descriptive terms from each of those abstracts, along with already done meta-analyses for over 1,300 of those terms and 150,000 coordinate-based co-activation maps. And a co-activation map is, in this case, for each of 150,000 voxels in the brain, they looked at all of the papers that report activation in that voxel and meta-analyzed all of those, showing which other parts of the brain that voxel is frequently co-activated with. And that's Neurosynth. So a little bit more about Neurosynth. Here is how it works broadly. I mentioned to you that it is an automatically populated database. And what Neurosynth does is it uses terms that are mentioned in the papers to classify what these papers are about. And then it extracts things automatically that look like coordinates. So it uses an algorithm instead of a human being to extract data from these papers. And then using those coordinate-like things, it can run a specific type of meta-analysis that results in the probability of pain given activation. And there we have it. So the important part is that this is all done programmatically, automatically. No human being had to do any reading or extracting of coordinates. It was all done with an algorithm. There are some caveats to this approach, of course. The first of which is that there are errors in coordinate extraction. And of course, the terms mentioned in the articles are only a rough approximation of what the paper is about, because a paper saying not pain would be the same as a paper saying pain, right? So even with those caveats, Neurosynth works really well. One indicator of success is that it is being increasingly used over the years. And the cool thing about Neurosynth is that it was the first database that was large enough to see which regions were preferentially involved in different cognitive processes relative to other cognitive processes. And you can use it to create a priori ROIs for analyses, which solves the problem of circular dependencies in analyses. You can also use it for functional decoding of regions in the brain, which we'll talk about a little bit later. But unfortunately, Neurosynth cannot answer all meta-analytic questions. For example, if we wanted to look at the difference between emotional pain and physical pain, that is not a question Neurosynth can answer because it requires a little bit more nuance than the bag of words approach that Neurosynth uses to extract the terms for each paper. Which brings us to the most recent automated meta-analysis platform on the scene, which is NeuroQuery. NeuroQuery, about 10 years after Neurosynth, another automatically populated database of brain extraction coordinates and terms from the papers. It includes about 13,000 functional neuroimaging papers, and on top of all of that sits predictive modeling methods. So NeuroQuery can indeed show us the difference between emotional pain and physical pain. So here are some examples of automated meta-analyses over the years. The first of which is a super cool paper. They did an independent component analysis of all of the papers in BrainMap, along with some resting state fMRI data, and found 20 brain networks that were the same in each. So that's cool because we're saying that these are resting state networks that are also broadly coherent across a whole bunch of different tasks in the BrainMap database. So you can see here, we've got our PAL, the default mode network in number 4, some visual networks, frontoparietal networks, central executive, et cetera, et cetera. And then a few years later, Andy Laird and colleagues did a decoding of those networks using the BrainMap database for some behavioral interpretation and clustering, which showed us that there are some emotional and interoception networks, motor, visuospatial networks, visual networks, and other cognitive networks. And then a few years later, we see Neurosynth used for a data-driven segmentation of human insula and interpretation of the roles of each of those segments. And we'll talk a little bit more about this process later, but it is one of the big highlights of the pros of automated meta-analyses. And finally, very recently, I just found this while I was making these slides, and I think it's super cool. People are using Neurosynth to create posterior distributions for Bayesian fMRI analysis. So here in A, you've got a typical GLM. In B, there is a Bayesian analysis with just like an out-of-the-box standard prior, and then C and D show different thresholds using a meta-analytically informed Bayesian prior, which is Super cool. So one of the reasons you might do an automated meta-analysis is specificity analysis. And that is you're going to use all of the maps in a database to interpret your statistical results using conditional probabilities. So using the example of that insula segmentation paper, they did a co-activation-based segmentation, looked at how different parts of the insula are connected to different parts of the brain, which gave them 3 different segments. And then they used these conditional probabilities with the whole neuroscience database to tell us what these segments are doing differently. And as I mentioned earlier, anything is possible with a manual meta-analysis. But the process of doing this manually would require you to extract data from all of the functional neuroimaging papers in PubMed, which again, infinite time, infinite energy. So another example of doing a similar approach is what Angie Laird did in that paper that I mentioned on the previous slide, that behaviorally interpreted those intrinsic connectivity networks using the rich behavioral data from the BrainMap database to look at the behaviors that each of those networks are doing. So when should you use automated meta-analyses? First of which, if you're going to do a domain general or a co-activation based meta-analysis, so meta-analyzing a broad construct like social cognition or co-activation based meta-analysis, again, these are looking at papers that report activation in a specific brain region and how those papers, like across the brain, what other brain regions are active or co-active. You can also use automated meta-analysis for hypothesis generation. So this could be making ROIs, choosing regions for functional MRS, etc. You can use them for functional encoding, which uses out-of-sample prediction using the published literature. You can also do functional decoding, like I previously explained, for interpreting your results. And Kendra is going to go ahead and talk about functional encoding and decoding a little bit more in the next talk, so stay tuned. Of course, there are limitations of automated meta-analysis. First, the search process is a little bit limited. Each meta-analytic database only contains a subset of the literature, and depending on how often they are updated, it might be a little bit out of date. The curation options are less flexible than reading each paper by yourself, which means a little bit less specificity in the paper selection from the search results. The data extraction process can be a little bit noisy, like I mentioned earlier, and the annotations can be kind of hit and miss depending on how they have annotated the papers in their database using terms or controlled vocabulary, etc. Which brings us to the best of both worlds, the semi-automated meta-analysis. So searching is easy because we are combining the powers of an automatically populated database with human curation. You can, depending on the method you're using, your search can begin with just neuroimaging papers that already have extracted coordinates, or a group of publications that include some extracted coordinates and some papers that haven't extracted coordinates yet. Curation is often facilitated depending on the platform, and it can be made more transparent and easy, and you don't have to use Excel, which is great. The extraction process, like I said, is mostly done. Depending on the platform that you're using, it might only search papers that have already had their coordinates extracted, or it might search all the papers and then return papers with coordinates extracted and without coordinates extracted. But either way, it saves you a lot of time. And of course, it is easy to annotate and share them if facilitated by the platform, for example, Neurosim Compose or the no longer active Metacurious, RIP. So platforms for semi-automated meta-analyses include the Neurosim Compose, which we're going to be using in the tutorial section of the course. And a few years ago, I was helping to work on a similar platform called Metacurious, which unfortunately no longer available. But on Neurosim Compose, you can see we've got, here's a study with extracted coordinates. You can add metadata, fix coordinates, add information, etc., and then use those in a meta-analysis. And we're going to go through that process in the tutorial section of the course with James a little bit later. So to recap, manual and automated meta-analyses serve different purposes and use different tools and have different drawbacks. Manual meta-analyses can be more nuanced and detailed, but they are often more time-consuming. Semi-automated methods use the best of manual annotation and curation along with an automated dataset population to speed things up. Automated methods are largely data-driven and can be hypothesis generating, but are also often very noisy. And please, please, please share your maps. I talked a lot about coordinates and coordinate-based meta-analyses in this talk, but it is better if you can do it with image-based meta-analyses, because as you might have heard earlier, coordinate-based meta-analyses are just trying to approximate what you would be able to do with an image-based meta-analysis. And the reason image-based meta-analyses are not more popular is because there just aren't enough images available for most topics. So please, please, please share your maps. Alrighty, so some acknowledgements. The reason that I get to be here today is because I had a wonderful doctoral advisor in Andy Laird who taught me a lot of what I know about neuroimaging meta-analyses. And I now have a wonderful postdoc advisor who allowed me to be here today talking about something that I no longer do. And I'd like to thank the Southern California Environmental Health Science Center, which is funding my trip to OHPM this year, along with all of the organizers and other speakers in this course who you're going to hear from throughout the day. And if you're interested in any of the papers that I mentioned, here they all are. Thanks so much.